Hello, my name is Patricia. I'm a postdoc member of the Berkeley Synchrotron Infrared Structural Biology program at the Lawrence Berkeley National Lab, and I will talk about multiscale insights into coralline algae holobionts and their mineral microstructure. Well, coralline algae are rhodophyta, a group of ancient and diverse life forms who had developed biomineral formation for millions of years. This feature is particularly important for our carbon sequestration research because today there's an urgent need to transfer carbon from the atmosphere into timely stable structures such as carbonates. Today, coralline algae populate vast coral areas and massive mineral beds. Their survival is threatened by global change because of warmer temperatures and ocean acidification scenarios. On the slide, you can see our study models, tidal pools of the Gulf of California. In these pools, there's a dominant coralline algae named Neogonioliton trichotomum. You can see the collector is holding it in his hand, and to the left, you can see a zoom of the little branch of this algae, which is as small as just few millimeters. In the bottom part, you can see some environmental characteristics of our sample scheme because we performed field work in winter where when upwelling is bringing cool and nutrient-rich waters to the tidal pools. Instead, the summer is characterized by low nutrient waters and warmer temperatures. There's a large amount of scientific literature related to coralline algae. By studying them, we have learned about evolution, biogeography, biogeochemistry, sedimentology, geology, and marine diversity, for example. But we just started to study them as holobionts or systems. This approach is helping to reveal unknown structural, ecological, evolutionary characteristics of these interesting systems. To study them as holobionts, we were wondering if is their vulnerability or resilience related to their microbial symbionts, such as in corals? What are the factors governing their symbiont ecology? And by looking at them as carbonate building systems, we were wondering what is the role of microbes in the biomineral precipitation? Because we know that microbes play an important role in other carbonate building systems. And what is their mineral structure and composition? And is their mineral composition or structure related to their vulnerability or resilience? And to explore these questions, we realized we needed a multi-scale assessment. So in our study, we integrated an environmental characterization that included nutrient status, 16S metabar coding, scanning electron microscopy, X-ray diffraction, and synchrotron infrared spectrum microscopy. In this slide, I summarized some of the most relevant findings regarding the prokaryote microbiota. In the ordination plot to the left, you can see we were able to clearly distinguish the algal microbiota than that of the surrounding water. But between seasons, the algal microbiota showed important differences related to certain prokaryote groups mainly. These differences were correlated, or the microbiota differences were correlated to environmental parameters. Particularly, temperature was the main axis of change, and secondarily, nutrient availability, particularly nitrate and silicate. These were performed. These are the results shown to the right up corner, and as you can see in these are performed by an MRT analysis based on the abundance of the microbial groups. And we described at family level the microbes more implicated in these seasonal differences. And interestingly, among them, we identified known halophilic and oligotrophic microbes. In this slide, I am showing some relevant findings regarding the microstructure. And to the left, you can see the mineral composition based on bulk mineral analysis of powder samples by X-ray diffraction. The main component is magnesium calcite, as has been reported broadly in the literature. But we also found aragonite and 2 iron-containing carbonates, siderite and ankerite. This last is a dolomitic carbonate. Interestingly, we found sylvite and bischofite in the summer samples, and these are evaporitic minerals known as potash salts. To the right, you see a lateral view of the algae branch, and the lower part is the hypothallus, and this holds this amorphous structure. And on top of it, there's a growing layer, the perithallus, marked in green, that looks much more organized or squared in this view. And there is also another thin, rough layer called the epithallus that coralline algae are able to shed as materials get attached to their surface. And I want you to imagine the growing layer without the upper crust from a top view, because I'm going to show you that in the next slide. And here to the left bottom, you can see this SEM micrograph of the perithallus hexagonal shaped cells. Some of these cells exhibit a content similar to spheres, as you can see. And on top, you can see the bright field microscopy of a single perithallus cell, which is more or less 20 microns in diameter. In the center, you can see synchrotron infrared chemical information. The heat maps represent spatially resolved infrared signals based on a single perithallus cell. Under the bright field image of the cell, you can see how we were able to detect a spatial segregation of 2 minerals, magnesium calcite and aragonite, in a scale as small as tens of microns. To the right, you can see the protein signals and the polysaccharide signals that occupy all the cell content. And by study with more detail the polysaccharide signals, we were able to identify an area of some tens of microns rich in sulfated polysaccharides. We were really surprised because we were hoping to detect a starch, that is the material that has been broadly described in the literature as the content of the algal cells. But instead, we found these sulfated polysaccharides, which have been also reported in red algae and are related to antimicrobial defense, but also to biomineralization. And in summary, we think this multi-scale approach has been useful to provide some clues to understand 2 different aspects. One is the seasonal microbiota plasticity, potentially related to adaptive responses of their holobionts, particularly to thermal and nutrient availability pressures. The second is a description of a strong microstructure, because we detected minerals known to be structurally resistant to dissolution in decreasing pH, particularly high magnesium calcite. The content of the magnesium calcite was higher than 18% in our samples. And we also found dolomitic carbonates such as ancherites. And besides finding halophilic bacteria, the presence of evaporitic minerals, both more abundant in the summer, suggest together halophilic microenvironments in the algal thallus. And we were also able to detect microscale mineral segregation and signals of sulfated polysaccharides that will guide our models of biomineral formation in red algae. And all this work is possible because of the effort of many enthusiastic participants. This is part of the master's thesis of Andrea Bautista. And this was an important effort of the BICA group of the Autonomous University of Baja California Sur. Some work done in the Institute of Ecology, the National Lab Langhem, and Faculty of Sciences and Marine Science and Limnology Institute of UNAM, and the Berkeley Synchrotron Infrared Structural Biology program led by Dr. Hoying Holman, and the participation of the very nice team of the infrared beam lines at the ALS at the Lawrence Berkeley National Lab. Thank you, and I'm happy to answer questions.